Hey everyone, today's micro seminar is coming to you from a slightly new date and time. So just to get it in before Thanksgiving, we're having it on a Monday today. I'm actually from my office, so that is also new. There's a big battle axe in the background, thanks to Mike Parker. But other than that, that is all for me. So I'd like to introduce today's micro seminar speaker, Dr. Luis Zaman. Zaman. I have actually Zaman, if that works. Zaman? All right. Dr. Luis Zaman, who has a pretty interesting history, and it's actually pretty cool that he was able to give this talk because it's fun for me to see. He does a very cool blend of both evolutionary biology and kind of computational stuff, and asks really cool questions from a variety of different directions. And so it's kind of awesome to see what he's going to show us today. Luis started off as a graduate student co-advised by the power duo of Rich Lenski and Charles Ofria at Michigan State University. After that, he did a postdoc with Ben Kerr. So it's, again, working in line of very awesome people. So postdoc with Ben Kerr at UW in Seattle for a little bit. And then for the last year or so has been a UM Collegiate Fellow. But good news, he is, I think, officially gotten the word that he's University of Michigan is going to give him a tenure track position and allow him to start his lab there completely. So he's going to start there, he says, in January, a full-time tenure track job, starting the Zaman lab. And so with that, I'll publicize up front that he says he's looking for all sorts of people, postdocs, grad students, anybody that's willing to come. Feel free to contact him. Obviously, his Twitter handle's here, but email and stuff is good too. But other than that, that's all I've got from me. So I'm very honored to be able to introduce Dr. Luis Zaman, and I will throw it to him.
Great. All right. Thanks, David, for that great introduction. And I'm going to turn on the screen sharing here, and you're going to see kind of a wild wormhole show up for a second. But all right. So like David said, I do a lot of mixtures of computer science and evolutionary biology, and most of my work is actually done more closely at that interface, looking at host-parasite coevolution. But I also do some other work on these kind of fun emulsion systems that I wanted to talk a little bit about. And this is where the rate yield trade-off project comes in. And to kind of describe what rate and yield mean, these race cars, or one of these cars that's a race car, kind of does a good job, in my mind, of painting the picture. So you can imagine both of these cars are made by Toyota. They're both gasoline combustion engine cars, but they're very different. One is very fast, but it uses a lot of gasoline in order to get the same distance that, say, the Prius does, ignoring the fact that that's a hybrid electric car. And the same process of having a rate versus yield trade-off occurs also with microbes. There's many different ways that we see it, but one of the most obvious ways is just how much ATP a microbe has to use in order to generate energy from some substrate. This is also true for microbes that have to express some transporters. There's energy involved in creating those, or active transport, where you're actually spending energy to get some of the substrate into the cell. So cells have these inherent sort of thermodynamic trade-offs built in. And the thing that really is interesting about them is that competition is what's driving evolution along this trade-off. And so competition comes in many flavors. You can obviously have competition for mates, for food. I'm not really sure what's going on in this picture. You can have competition for pollen if you're a bee. The flowers are competing for pollinators in that case. Dogs that we have in our house are competing for affection. They're competing for treats. Competition is literally everywhere, and microbes also compete. And so in this little graph, in this little figure, there's two different populations of microbes I'm depicting by the blue and the red circle, and 6 different glucose molecules here in the environment. And you can imagine one very fast microbe that can go and get that glucose really, really quickly, and a slower type that is maybe not spending as much energy to get the glucose, but can use it more efficiently and grow to a better density per unit glucose. But if these two things are sharing resources, the blue type, even though it's more wasteful, is able to go around, get all the glucose, and grow, while the red one is still sort of working its way around getting just one glucose molecule. And we can do microbial competition experiments that are just basically that, except on a very large scale, where we take 2 different populations of bacteria, mix them together, and just let them grow for one day and see which bacteria strain wins. And we can do this because we have these nice neutrally labeled bacterial cells where one produces a red pigment on a particular type of indicator plate and the other one doesn't. And so we can mix these two together at different, so in this case it's saying ancestral and evolved, and that'll come back in a second, but we mix them together at the same ratio, let them grow for 24 hours, and then look at how different the ratio is after that time period. Now what's cool is we can do that over a very long period of time where we've actually just transferred these back microbes into fresh media every 24 hours. And this is an experiment that some of you probably know, most of you probably know from Richard Lenski's lab, that's been going on for a very long time now. But what this lets us do is basically go back to any previous time point and revive the bacteria and do these competition experiments. And what that gives us is a measure of how fit the bacteria are getting. And the fitness in this case is really that competitive ability that I was trying to get across with those two red and blue populations going after the glucose. This is really a measure of how much faster you are at getting the glucose and how much more bacteria cells you produce in a given time, because these are all sharing resources in one big mixed population. Now, this experiment has been going on for a long time. You saw on the graph it was 60,000 generations in that figure. They're actually up to almost 70,000 generations for all of the replicate lines. Some of them are a little lagged than others, but this experiment has been going on for a long time. It just celebrated its 30th birthday. I was in the lab when it celebrated its 25th birthday, and there are still pictures going around from its 16th birthday, including the little car there signifying that it got its learner's permit. And you can see the little hats on each of the 12 flasks up in the top left here. Now, if we put that into perspective just on how long this experiment's been going on, we can take kind of a rough approximation that maybe 15 years per generation is on the order of what we might expect human evolution to, or human reproduction to occur on. This is obviously a very big range, and that's just one number, but if we did that, it would range it's about 900,000 years ago. So we're talking really long time. A lot of evolution has occurred, and this is a really great resource. Now, remember that this is all happening in a batch culture. So what we're really selecting for are those blue microbes, those ones that are going and getting the resource faster than the other types. If you get all the resource before all of your competitors do, then there's no resource left for the competitor, and you're just going to grow. You're going to beat them. So that's what batch culture is selecting for. Now, what that creates is basically this rate yield trade-off. So we have the blue type that has a high rate but a low yield, and yield here, I'm talking kind of abstractly, but we'll get into a specific definition in a second, versus this red type that was much slower but was able to grow to a higher density per unit glucose. But if we dropped a wall between these two pools of resources, and now we didn't really care about, there's no competition anymore, right? So it doesn't matter how fast you get the resource. They're both going to go after the glucose at their own rate. But the red type, because it grows more per unit glucose, is going to end up at the end of eating all of this resource to have a higher population size than the blue type. Now, one way we put walls in microbiology experiments is using these 96 well plates. Now, I think most people that are watching this are probably familiar with these, or either love them or hate them. It depends on how many of them you've used in your life, I think. I am more on the side of hating them now, but that's okay. But you can imagine if we want to put just one cell into each one of these wells, first off, that limits us to 96 wells in a given plate. But also, you're dealing with very small numbers, and you have to kind of rely on a Poisson distribution, where you're not really, if you shoot for one cell per well, you might get some wells with 5 cells, you might get some with 0, and that ends up with a lot of waste. And really, if you really only want one, you should be shooting for something more like 0.3 cells per well. And there's a lot of empty wells in that case. So it's a lot of plastic, it's a lot of waste, it's not super efficient. So what we decided to use was this kind of new technology where you basically take a tube of bacteria. Here's the culture, just normal LB media, for example, with some bacteria cells. You add this oil and a surfactant and vortex this tube for 1 minute, and what you end up with are just millions, hundreds of millions of these little tiny droplets of media surrounded by oil. So the surfactant is changing the chemistry so that the water is actually the thing that becomes surrounded by the oil. And that gives us these very tightly independent bubbles that cells can live in. And then we can let the cells grow in this media, incubate it just like you would any other culture. And we can break the emulsions by adding a demulsifier. And then, yeah, that's pretty much the standard protocol of working with these emulsions. And a lot of this work is thanks to Herwig Bachman, who's now at the NIZO Food Research Center at it's in the Netherlands. And he is an incredibly nice guy, and I highly recommend checking out some of his papers. He actually went through and figured out how to synthesize all of the chemicals that are required to develop this emulsion system in batch, and also figured out which of the commonly consumer grade chemicals you can buy from different chemical supply companies you can use to do these emulsions. So it's a really great resource. And one of the really interesting things about this system is that the chemistry and the technology that made these emulsions possible actually came out of the cosmetics industry. So for a long time, cosmetics, especially say like moisturizers, facial moisturizers, have been relying on water and oil emulsions to deliver the products. And so a lot of the benefits that we're getting as a scientific sort of direction using these emulsions have come from these cosmetic industries. So it's just a fun kind of side story. But this is what they look like under the microscope. This is about a millimeter long slice of an emulsion. And you can see there's a lot of variation in the droplets. And that's okay because there's a lot of droplets. And so we shoot for an average droplet size of about 50 microns. And that gives us enough volume for about 100 to 1,000 bacteria cells, depending on their size. There's a big range of microbe size, so that's a big range, obviously. Now, we can put some fluorescently labeled cells in here. And so here I'm showing that same size of a field of emulsion droplets, but now we're aiming for a very low density of cells within each droplet. So here, I believe we use 0.3. So on average, only 1/3 of a cell should end up in a droplet, but obviously there's single cells. You can't divide them. So you end up with a lot of empty cells and some droplets that have more than one cell. So you can see up here, for example, there's a droplet that's got both a red and a blue type. We mix 50% of two different fluorescently labeled cells. But some of them are single colored. Most of them are single colored labeled. And even here, for example, you can see two droplets that are right next to each other, but are differently labeled. So there's no cross-contamination between droplets. And because we have fancy confocal microscopes, we can make cool 3D images of these things. Now we can zoom out a little bit and go for a high density emulsion. And here, instead of 0.3 cells, we're shooting for 3 cells per droplet. And now you end up with a lot more droplets where both cell types are in. So this one in the middle, for example, is pretty easy to tell. You still have some droplets that are kind of uniform in color, probably. It's harder to tell at this scale, but it's still expected if you have a Poisson distribution where you're shooting for 3 things. Sometimes you'll get 1, sometimes you'll get 2, but those 2 might be the same fluorescently labeled cell, right? So there's still a variation in the kinds of cells that or the kinds of populations we're ending up within each of these droplets. But we get these kind of really cool videos of each of these little tiny subpopulations that we can watch, and I can sit and watch this for a really long time, but I won't. So again, we have this frozen record of rate adaptation because these things in the long-term lines that have been evolving for 60,000 generations have been evolving in a batch culture where the sugar is available to every single microbe at the same time. There's no separation of population. There's no subpopulations in these. So what we can do is go back and look at different time points along this, because these are all in the freezer. We can revive them. So we have the ancestor and let's say a 2000 generation clone, but we can also, and we can do these competitions where we mix them together and we see how much more fit the evolved type is than the ancestor. So that's what the evolved and ancestor labels were from that original figure. But we can also do that across the whole transect of time. So we can go out to 20,000 generations or 60,000 generations. And now we could go out even to 70,000 generations. And one of the things that the long-term line is really good at is reminding you how long you're taking on your project, because those numbers keep ticking along. And when we do this competition, we can do this competition now in emulsions. So the same process of mixing the 2 bacteria strains together, putting them in emulsions, then letting them grow, breaking those emulsions and plating them like we could do with just batch culture, we can do also with emulsions. And so when we do that, there's only 2 data points here, so don't get kind of confused by the 1 and the 2. There's no data at those 2 points. We can look at how the proportion of the evolved type that's in this competition changed over that 3-day period of growth. And so 2,000 generations here, we have 3 different replicate populations. The 3 replicates are true biological replicates, so they're different evolved strains of the long-term line evolution experiment. So in the LTEE, there are 12 replicate lines. We picked 3 of them, and those 3 are the ones that we're competing here in an emulsion. So at 2,000 generations, 3 different biological replicates, we see that the proportion evolved in the high emulsion, so this is what this little icon's saying, that there's more than 1 cell per droplet, we're shooting for 3, that the evolved type is increasing. Now we can do that for all of our different time points, the 2,000, 20,000, 60,000, and what you hopefully see is that the variation in how the proportion of the evolved types is getting much more tight. There's more signal that the evolved types are winning in these high density emulsions, and this is what we'd expect, right? You have a droplet with multiple cells now, and those cells are competing for a shared pool of resource. It's not the full pot of resources like you would get in a flask, but there's still multiple cells per droplet competing for a shared pool of sugar. So the types that have evolved to get the sugar really, really quickly are going to win, and that's what we see. But if we go to a low proportion or a low density emulsion here with this low icon where we only expect 1 cell per droplet most of the time, we actually see the opposite. And this was a pretty surprising result in that these are bacteria cells growing in an environment that they've evolved for, in some cases, for 60,000 generations. But now we're just kind of isolating them and keeping them away from using all of the resource all at once. And the ancestor is actually winning. So the ancestor of the experiment is what we're competing all of these against. So this was really cool. And we wanted to ask, how does adaptation to yield? So this is sort of, if we evolve strains in the emulsion now, how does their previous level of rate adaptation affect how good they can evolve or how fast they can evolve? So you can imagine we have this rate yield curve. Now we've shown that these 2,000, 20,000, 60,000 generations follow along this curve. It may not be so perfect as this, but you can just kind of imagine. And we want to push these populations now. Let's select them for yield as opposed to rate, and we could have different expectations. So is it that because the 2,000 generation clones have only evolved for a little while at rate selecting environments, do they evolve faster towards yield? Or is it that the 60,000 generation types, because they're just so much better, they've been evolving for so long, can sort of jump over all of these other populations? These are sort of ongoing questions, and there's good explanations for why you might expect any one of these cases to be true. And so this is kind of what I call a perfect experiment, where basically any result is a good result. And yeah, it's always something good to shoot for. So to describe how we do these evolution experiments in the emulsion, it's actually pretty simple. We start with the same process of creating an emulsion, but all we have to do is go from the end of one day or one transfer and take what we broke out of the emulsion, dilute it, and start again. And so we can dilute to different levels depending on how many cells we want to shoot per droplet. But this is basically the entire process. And it sounds really easy, but there's a lot of technical challenges involved. And fortunately, Katrina Van Ray she's finishing up her PhD with Ben Kerr right now, and she took over this project when I left and has been doing a really fantastic job getting the evolution experiment set up. And she gave a talk about some of the data that I'm going to show in the next few slides at Evolution, which is also online. And I'm guessing there could be some sort of link put in the YouTube comments or description, but check that out. She does a much better job describing this than I do. So for this, what we're going to look at is only evolution from the 60K, the 60,000 generation clones. And these have been evolved in low emulsions. So low density emulsions, these things should be evolving to get better at being yield adapted, better at consuming the resources slowly but more efficiently. And so just looking at this data again, this is basically just repeating some of the data we saw already. If we put them in a high density emulsion, the 60,000 clone does much better than the ancestor in competition. Right? So that's expected. If we go to low density emulsion, what we see is the ancestor does equally well, but the evolved type is actually underperforming. So this is, again, reversing that rate adaptation that we saw through the long-term lines, but because these things are privatizing their resources. And A, B, and C are three different clones that evolved during, I can't remember now, 100 transfers, I believe, maybe 90 transfers of low emulsion evolution, starting from 60,000 generation evolved clones. And so when we look at A, if we compete it under high density emulsions, it doesn't do very well, right? It's quite low. So this is a big difference, right? So 60,000 to A are the same strains, just this is after evolving in the low emulsion environment. So it's losing quite dramatically in the high competition, and it's definitely improved over its 60,000, and this is sort of the ancestor now of this evolved clone. Now we look at the other two, and we see that they actually are improving in both environments, sometimes really dramatically. And so this is kind of a confusing result in one sense, but it also is sort of what we expected also. And why I say that is because we think of this trade-off as just one sort of line, but really there's kind of multiple dimensions to this trade-off. And it's not necessarily the case that just because we don't see these results, we don't see that they're sort of losing out on one that they're not losing out on one dimension of this, that we don't expect there to be a trade-off anymore, but what we could be seeing is actually evolution off of this trade-off. Obviously, there's a lot more work that needs to be done in order to verify this, but this is sort of an exciting direction in trying to figure out now what those cells are doing in order to break free from this trade-off that is sort of so thermodynamically expected. Now, just wanted to show one other video that Katrina has produced. So about halfway through the experiments, the Kerr lab got a Coulter counter, which has helped us to measure stuff and also perform these transfers a lot more quickly. And it also let us get these really cool videos. So I'm going to show two videos at the same time. So one is in this high density emulsion, and one is in the low density emulsion, and what we're looking at is the size distribution of cells in that transfer. So each frame of this video is a different transfer, and what we can see is sort of how the distribution of size of cells changes. And so one of the things we might expect if you have selection to produce more cells is to make more smaller cells. And that's exactly what we'll see, to give away the punchline, but so that you see what you're looking at. But at the same time as we see in the low emulsion smaller cells evolve, we see larger cells evolve in the high density emulsion. And this is kind of consistent with what we see in the long-term lines, that the more rate adapted cells are actually bigger. And there's some complications with these videos, and I'll let you find that out by watching Katrina's presentation. But some of the cool stuff is that we're actually starting to really dig into what the difference is how the cell size is really playing a role in this trade-off. And that's work that's also going on with Rich's lab. Nkrumah Grant is working on this, as well as looking at how anaerobic evolution, or how evolution in aerobic has affected anaerobic adaptation. So there's a lot of fun stuff going on there too. Now, another project that we were pretty excited about was this idea that we could look at the entire metabolic pathway of cells using these whole genome models and really figure out where the adaptation was occurring that allowed energy to be used in a sort of more efficient or more rate adapted way. And so this is work that was done with Chris Marks, who's been very successful at this flux balance kind of models. And one of the really cool things you can do is you can analytically solve these models, asking what's the best pathway that the cell should use given some environment. And then you can go and test what are the pathways the cell's actually using by giving them radioactive carbon labeled glucose, and seeing where those particular radioactive atoms end up on the downstream metabolites. And so this is a really powerful technique. And one of the cool things is most of the strands that we're using have actually already been analyzed, but from the long-term lines. And so we can look at on a space, this is sort of just a multidimensional space where we have our ancestor and where the evolved strains are. And we can also look at where the predicted optimal is given this flux balance analysis. And what we see is that evolution actually moved away from the predicted optima. And this was, kind of disappointing, but at the same time, what people realize is that the flux balance analysis, the way that the mathematics works out is that you're optimizing for yield and not rate. And so one of the expectations is maybe if we evolve these things in emulsions, we can see evolution actually move back towards the predicted optimum. And maybe we have a little bit of a sense of that that's happening because the ancestor is obviously doing better than the evolved types in the emulsion. So this is ongoing. One of the complications, though, is that we started doing some of this metabolic flux analysis, just roughly characterizing what are the byproducts that show up in the media. And it turns out that it's a lot of things that are more associated with anaerobic fermentation. So droplets are definitely different. So we're sort of learning more about the system as we go along, but there's a lot of interesting questions still to be done. And that's actually it. And I think I went very fast. But I wanted to also thank Ben Kerr and Rich Lenski since they weren't featured in any one of those images. And I'm happy to talk more about all of these things online or offline. So that's it. And I'll close out of here.
Yeah, let's do the matrix thing again.
Yeah, definitely.
That was awesome, man.
Thanks.
Vaughn's here too.
Oh, great. Hey, Vaughn.
I have a question. There's nothing I see in Twitter yet. So if anybody has any Twitter questions or anything, just tweet them in, and I will keep looking for them. Question I have: so in the droplets, like, does anything go between them? So you have independent populations, but are there metabolites or like DNA or like plasmid transfer? Anything? Does that happen, or is it really truly separate?
So as far as we can tell, they're really truly separate. I think oxygen and actually, we know methane travels between them pretty freely, but other than gases, I don't think things travel super freely between them. But it is something that we have to keep an eye on, and probably I've got some ideas for experiments that we could do to test whether, say, viruses go between them. Something with sort of maybe a lipid layer might transfer between them. So maybe some of the single-stranded RNA viruses.
Cool.
Give it a shot.
Yeah, and then so the other question I have about that is that in terms of, so you're setting this up as a rate versus yield and changing the densities within the droplets, but you're changing like population size and bottleneck points and stuff too. So it's changing the population genetic parameters, which might change how kind of the availability of what mutations are there and stuff. So how do you take into account that, or how do you basically address that?
Yeah, that's a good question. So one of the ways is really by comparing the high emulsion to the low density to the high density emulsion. So that gives us a lot of the changes in just the chemistry of their environment because of the emulsions that we can compare directly. But there's still a big difference in population size there, and then the dilution's obviously different too. So what we've been doing is sort of characterizing while we do the transfers, what the bottlenecks are, how big the populations are, and then adjusting how many transfers we do of each when we compare them to adjust for the sort of expected number of doublings. Cool.
And is that all published yet, or is Katrina still like working? Is it?
No, so Katrina's still working on it. So you saw the evolution talk and know that there was.
Yeah, I linked to it in the tweet too, so I'll put that on the YouTube. I'll put it in the YouTube comments or the box going forward. But yeah.
Yeah, so you know there were some complications. Some of the things that we thought were evolution were actually contamination. The stuff I showed you was those were verified, not contaminants. Those were de novo mutations, de novo evolved strains. But yeah, it raises some interesting questions on how to wrap this up neatly. And so there's some pretty fun experiments that we're kind of following up on. So some people, Tim Cooper, the other Cooper, has a nice library of all possible combinations of the first 5 mutations that arise in the long-term evolution experiment. So we can put those in emulsions and see how each of those mutations affects rate and yield independently.
Cool. I would almost expect that because you get like crossfeeding and stuff evolving pretty early too in the long-term lines, like that could be weird in terms of like weird crossfeeding interactions within the, if there's a, there's a weird threshold there where it goes from rate versus yield, and then that ends up being selecting for a bunch of different evolutionary dynamics there, but that'd be cool.
Yeah, so one of the reasons we ended up starting off with this emulsion stuff in the first place was for this bios. There's a synthetic biology lab that we work with a lot, Eric Clavens, and they have a strain that they've engineered that stochastically switches. So it's one genotype, but it stochastically switches into either a producer or a consumer of, and the producer produces a cellulose degrading enzymes, so cellulases, and the consumer just eats it. So it has the right cellobiose pathways. But what that means is you can have one cell in a droplet and it'll diversify as it grows into these two different types. And so if there's cellulose in the media, then you can get these cross-feeding interactions and select for basically cooperation between these different strains. And I forgot to mention the producer has to lyse itself in order to let go of these enzymes. So it's suicidal altruism.
Cool. Is there like a, sorry, I'm monopolizing this. I have many more questions. But is there like a natural analog in terms of like gut microbiota and stuff? So it seems like these little small populations and kind of droplets, it might, it almost is like, you get little crypts and stuff in the gut and different places where there's these really small populations that can independently kind of evolve from what else is out there. So it seems like a decent analog, at least in terms of when we try to design these evolution experiments versus kind of what might be happening in real life. But
yeah, definitely. I think so. I don't know as much about the gut, but particles in the ocean, I think, tend to be a really good analogy, especially because they're kind of like little droplets. They're not encapsulated, but you have biofilms growing on all these little particles, and there's transmission between particles of different bacteria cells. I'm sure Vaughn has more to say about that.
Cool. All right. Well, let me check Twitter one more time. Not seeing anything. Apologies if I missed it. But other than that, man, so that was awesome. Like I had to tweet out like this is awesome tweet out there for just describing the whole system. But so thank you very much to everybody out there. Happy whatever holidays you celebrate going forward. We were yeah, I think there's no micro seminar scheduled until March. So if you guys have anybody, or if anybody wants to present, feel free to get in contact with any of our organizers. We've got a few open dates coming up, and I'll be publicizing that on Twitter too. So get in touch with us too. But thank you again, Luis, and good luck in January when you you realize how much of the sausage is made behind the scenes that you never realized before.
Oh, great!
All right, man. Thanks a lot.
Thank you. Take care.